Hi everyone, I am J.G.DHARANY, Assistant Professor of Biochemistry, A.D.M. College for Women, Autonomous, Nagapattinam. Today, we will discuss the topic is strain technique. Straining. This is the introduction of the strain technique. Straining is a technique used in microscopy to enhance concerns in the microscopic image in biology and medicine to highlight structure in cell population or organelles within individual cells. Dyes are colored organic components that can selectively bind to tissues. Most modern dyes are synthesized from similar organic molecules, usually benzene or one of its derivatives. This is the strain structure. Most simple organic compounds such as alkalines, benzenes and alcohols are colorless to the human eye but will absorb light outside the visual spectrum. Benzene, for example, absorbs strongly in the UV region of the spectrum but appears water white to the human eye. This is the staining process. The actual staining process may involve immersing the sample, for example, before or after fixation or mounting in dyeing solution followed by rising and absorption. Many dyes, however, require the uses of moderate. For example, a chemical compound that reacts with the stain to form an insoluble colored precipitate. When excess dye solution is washed away, the moderate stain remains. Stains are generally prepared largely as aqueous solution. This is also staining process. Some cases stock solution are prepared in alcohol and are diluted with water as needed. Since alcohol removes the stain, pure alcoholic solution should not be used. Staining solutions are prepared to contain low concentration to stain rarely exceed 1%. This is the factor of staining. Factor which affecting staining. We contain 9 points. First point we see solvent, alcoholic or aqueous solution. Second point is low or high temperature during reaction. Third one is simple or multiple combination of dyes. Fourth one, the covering power of the dye. Fifth one, the time is called as period. The time the dye acting. The types of the tissue and the fixation used. Seventh one is the type and thickness of the selection. The eighth one is the temperature and applied during dyeing paraffin selection of the slides. This is the classification of stains. It contains 3 types. First of all, we see according to their chemical composition such as organic and inorganic. Organic are hypoxylene stain, caramine stain, aniline stain. Then second one is inorganic. Classification of stain second type is according to their chemical behavior: basic, acid, neutral. Classification of stains is called third type. Histological stains are nuclear, plasma or general stain, specific stain, empirical. This is the classification of dyes. Dyes is also called as stain. An acid dye has a negative charge. For example, benzoin, rose bengal and acid. Since there are negatively charged bind to positively charged cell substance. The dyes stain better under acidic conditions where the proteins and many other molecules carry on positive charge. A basic dye carries a positive charge. For example, methylene blue, basic purple, crystalline violet, melatonin green, sapphire. Basic dyes bind to negatively charged molecules. like nucleic acid and many proteins. This is also classification of dyes since the bacterial cell surface are negatively charged. Basic dyes are most often used in bacteriology. Basic dyes are normally available as chloride salt. A neutral dye is a complex salt of a diacid with a dye base. The dyes used in bacteriology have two features. First of all, we see first feature. They have chromosome groups which double bond that gives the dye its color. Second point, they are bind with cell by ionic, covalent or hydrophobic bonding. Most of the dyes commonly used in microscopy are available as certified strains. This is the uses of common dye names. Dye manufacturer usually use the dyes they produced common names such as isonine or Congo red rather than their full chemical name. The full chemical name of Congo red is 3,3-biphenyl 4,4-dibenzyl bi-4-amino 1-naphthol sulfonic acid disodium salt, which is common name is Congo red. More of the problem is the fact the different dyes can be produced by different manufacturers under the same name. For example, a dye called light green is usually considered. An acid dye is histology and used for staining connective tissue. Buying the wrong dye can totally alter the result of the staining method. In vivo staining and in vitro staining. First of all, we see in vivo staining. In vivo staining, called as intravisual staining, is the process of dyeing living tissue in vivo means in life by causing certain cells or substrates to take on constructing colors. They form morphology or position within a cell or tissue can be readily seen as steady. In vitro staining involves coloring cells or structures that have been removed from their biological consent. Certain strains are often combined to reveal more details and features than a single strain alone. Those strains excluded by the living cells but take up by the already dead cells are called visual strains. Those that enter the strain living are called supravital strains. For example, new methylene blue and brilliant crystalline blue are ready circulate staining. These stainings are equivalently toxic to the organism, some more to than others, partly due to their toxic interaction inside a living cell. Note that many strains may be used to both living and fixed cells. For example, for staining, types of staining: simple stain, compound stain, indirect stain, direct stain, and progressive stain. First of all, we see simple stain. It is a staining solution contains one dye. Compound stain. This staining solution is composed of more than one or more dyes. Indirect stain. Staining needs a moderate to work. Direct stain. The stain works without adding a moderate. Progressive stain. When the different elements in the tissue are colored in sequence and at the correct time, differential coloration of tissue and achieved. It is a selective stain. The selective stain dyes are more than one substrate in the same color, but easy to identify these substrates. You want to dominate either by morphology or by size. Specific stain. When the stain acts only on the certain content of the cells or tissue or has a little or no effect of the elements. This is the impregnation. Some elements are demonstrated by method known as impregnation technique. These solutions used in the impregnation are the not dyes, but are solution of metallic salt. Cytological strain. This is the last strain, which are used to demonstrate of minute structure in the nucleus and the cytoplasm of cells. This is the structure of the staining of bacteria. Types of staining techniques is divided into two types. One is simple staining, other one is differential staining. Simple staining is a single basic stain. Differential staining is a two decoloration agent. This simple staining is used for visualization of morphological shape and arrangement. This differential staining also divided two types. One is identification process, then second one is visualization of structure. This identification of techniques contain two types. One gram staining and the other one is acid fast staining. This visualization staining structure of two types: spore staining and capsule staining. This is the common biological stain. There are many types of staining techniques. All of these dyes may be used by fixed cell and tissue. First of all, we see gram staining. Gram staining is used to determine gram status to classify bacteria broadly. It is based on the composition of the cell wall. Gram staining used crystal violet to stain cell walls. Iodine as a moderate to mark all bacteria. Gram status is important in medicine. This is also gram-positive bacteria process. Gram-positive bacteria stain dark blue or violet. Their cell wall is typically rich with and lack of secondary membrane and lipopolysaccharide layers found in gram-negative bacteria. Gram-negative organisms appeared red or pink because there are contact stains. This is the structure of the gram-positive and gram-negative bacteria. First of all, it contains 5 processes: fixation, crystalline violet, iodine treatment, decolorization, and finally we content stain with sapnine. This is the second technique, negative staining. A simple staining method for bacteria that is usually successful. Even when the positive staining method detailed below fails, is to use a negative stain. After dyeing, the microorganism may be visualized in bright field microscopy as lighter insulation when concentrated against the dark environmental surrounding them. This is the third type of stain, Nickel-Nielsen stain. This is used to stain species of Macrobacterium tuberculosis that do not stain with the standard laboratory staining procedure like gram staining. These stains used are the red-colored carbon chosen that stain the bacteria and a counter stain like methylene blue. This is the fourth type of staining, Hemotoxylin and isolin stain protocol is used frequently in histology to examine thin selection of tissue. This stain blue cell nuclei with isocyne stain, cytoplasm, connective tissue and other extracellular substance pink or red. When this stain is done by this machine, that suitable difference of eosinophilia. Hemotoxin stains the cell nucleus and other acidic structure such as RNA each portions of cytoplasm and the matrix of the cartilage blue. This is the structure of the hemotoxylin stain. This is the fifth types of the stain, arginine orange. Arginine orange is also called as AO stain. Is a nucleic acid selective fluorescent cationic dye useful for cell cycle determination. It is self-renewable and interact with DNA and RNA by interaction of electrostatic alteration. This is the sixth type of strain, crystalline violet. Crystalline violet, when combined with a suitable moderate strain, will called purple. Crystalline violet is an important component of gram staining. This is the seventh type of strain, ezone. Ezone is the most often used as a counter strain to hematoxylin, impairing a pink or red to cytoplasmic material, cell membrane and other extracellular structures. It also impacts a strong red color to red blood cell. Ezone may also be used as a counter strain in some variant of gram staining and in many other protocols. This is the eighth type of strain, ethylene bromide. Ethylene bromide is intercatalyzed and stains DNA, providing a fluorescent red-orange stain. This stain may also be in conjugation with arginine orange. This is also called as AO stain. Is viable cell counting. This EB and AO. EB means erythrithium bromide and erythrit orange combined stain causes late cells of fluorescent green, which list of cells retain the detective red-orange fluorescence. This is the ninth types of strain, acid fusin. Acid fusins may be used to strain collagen, smooth muscles or mitochondria. Acid fusin is used as the nuclear and cytoplasmic strain in Mallory trichrome method. Acid fusin strain cytoplasm in some variant of Mason trisomes. This is the 10th types of strain structure. Hemotoxylin. Hemotoxylin in North America. Hemotoxylin is a nuclear strain used with a moderate hemotoxin strain nucleic blue, violet or brown. It is most often used with isocyane in H and E. H and E means hemotoxylin and isocyane strain. One of the most common procedure in histology. This is the 11th types of strain techniques process called iodine. Iodine is used in chemistry as an indicator for starch. When starch is mixed with iodine in solution, an intensely dark blue color develops representing as starch or iodine complex. Iodine is also used as moderate in gram staining. For example, is one component in the staining technique used in morphology or microbiology. It enhances dye to enter through the pore present in the cell wall or cell membrane. This is the 12th types of strain technique. Melanine green. Melanine green, also known as diamond green B or Victoria green B. This melanine green can be used as a blue-green counter strain to saffron in staining techniques for bacteria. It also can be used for directly stain pores. Then this is the 13th stain technique, methylene blue. Methylene blue is used to stain animal cells such as human sick cells to make their nucleic more absorbable. Also used to stain the blood, spleens and used in cytology. This is the 14th types of stain technique, neutral red. Neutral red stain methyl substance red. It is usually used as a counter stain in combination with other dyes. Safnanin. It is the 15th types of stain technique, safnanin stain. Safnanin or safnanin O. It is a nuclear stain. It produces red nuclei and is used primarily as a counter stain. Safranin may also be used to give a red color to collagen. This is the last other chemical used in electron microscopy staining includes ammonium molybdate, chromium iodide, carbohydroxide, ferric chloride, hexamine, indium thiochloride, lithium nitrate, lead acetate, lead citrate, lead nitrate, thiosemi carboxylate, uranyl acetate, uranyl nitrate, and vinaitin sulfate. Thank you.